Welcome back to the Saving Sapiens podcast. Prion diseases have been one of the most mysterious and fascinating neurological phenomenon to be studied by medicine. They have resisted diagnosis and treatment. And today, I am excited to be talking with Dr. Kariem Ezzat about a brand new finding that may push out the old model of prion disease and perhaps will hold the key to making this type of disease treatable for the first time. Welcome to the podcast.
Yes, thank you very much, Matt. Thank you for having me.
So, introduce for us the fascinating and mysterious world of prion diseases and why they, for quite a long time now, have looked different than other neurological diseases, and then talk to us about why they have been untreatable.
Yes, I think we can start by Robert Koch, who devised the microbial theory of infection in the late 1800s, early 1900s. So I think, in my opinion, he's one of the most influential scientists of all time. He managed to show that microbes cause a lot of devastating human diseases. So he discovered cholera, and while he was alive, many of his students discovered like that TB causes tuberculosis. The TB bacteria causes tuberculosis, and so of course he was in this competition with Pasteur related to the microbial theory of infection. But within a very short time, the world understood that yes, microbes cause a lot of horrible human diseases, and they are the causes of infection, and especially initially bacteria, then viruses were discovered not so long after, in the 1930s, and then bacteria and viruses were seen as the causes of many of the epidemics and endemics in human populations. Fortunately, in the 40s and the 50s, antibiotics were discovered, and the rest is history. This was one of the most influential medical victories humanity has ever achieved. It gave us, it extended the lifespan. It nearly doubled the average lifespan of humanity by preventing the majority of juvenile deaths of children. And remember a time not more than 100 years ago that if you scratch your hand on a flower thorn or something, you can just die of infection. There was nothing to help you. So imagining now we live in the world everybody when they get an infection, it's a matter of days, if not a maximum weeks. They will just get an antibiotic and they will be free of that infection. So I think this was an incredible victory for science, and incredible discovery. But so many things were, many of the things that transmissible diseases could be explained by bacteria and viruses. You can find that TB, you can find it, or you can find the agent. So except for one mysterious disease, which was infecting sheep, it was called Scrapie disease. So Scrapie is kind of a neurodegenerative disease of sheep. It spreads in the herd, and it causes the sheep to scratch their fur to the fences, and that's why they scrape themselves. That's why it was called Scrapie. It was very annoying for the farmers in Britain who had to kill much of the herd if it has creating infection. So people naturally at that time, 1950s, 1960s, the microbial theory of infection is very established. And everybody was looking for what is the bacteria or the virus causing this infection. The problem was that they couldn't find any bacteria or virus causing this infection. And the methodology they used was basically to get the brain, because it's a brain disease, so they get the brain from the sheep and homogenize the brain, so make a brain homogenate, and then take this brain homogenate and inject it into another brain of uninfected animal. And after some months, this animal gets similar symptoms to the infected animal. And this was the model by which Scrapie was studied. And I think it was this methodology is one of the roots of the fundamental problems now we have in amyloid diseases in general, or neurodegenerative diseases in general, because it's not a method by which normal infection happens in nature, like taking a brain homogenate and injecting a brain of another animal never happens in reality. So much of the interpretation of these results was taken to indicate that yes, some agent in the brain of the infected sheep induces the disease in the resilient sheep. So it has to be an infection, and when they tried, when this homogenate was boiled or subjected to UV radiation, stuff that are supposed to kill any bacteria or also to kind of destroy any DNA from being able to replicate, and still the recipient sheep can get the disease. So this is when in the 1960s, 70s, and 80s, people were starting to think that this is a completely new way of how infections spread, that maybe it's not based on DNA, because bacteria and viruses have DNA, and if you boil this homogenate, you destroy living bacteria, and if you use UV, you destroy DNA. So if this homogenate can still induce disease in the recipient sheep, it must be something else that is not based on DNA. And this is where the Prion hypothesis was officially born in 1982 by a paper by Dr. Stanley Prisoner, who suggested that it is a proteinaceous infectious agent, and it's a protein that kind of drives other proteins to misfold and cause disease. And this was, I'm sorry if you're thinking so much about this history, but the thing is that this was the context of which this hypothesis was developed. It was not based on structural understanding of what the Prion agent is. It was just a way to explain why injecting brain homogenate from one animal to another can still lead to disease without after destroying all the possible presence of nucleic acids in this homogenate. So it wasn't based on structure. It wasn't like the discovery of DNA templating, for example, and when I think the paper by Watson based on the data from was going Franklin, that was based on X-ray structures, very detailed mechanism, very detailed template mechanism. A has to be with C and C has to be with G. It's the only hydrogen bonding is possible between these two pairs. This way, it's a very exact mechanism of how calculating and protein DNA replication works. For the protein only replication, it was just a way to explain the difficult to explain fact that you can still induce disease with the apparent absence of active DNA.
So the initial finding of Scrapie in sheep was kind of the first flag that there might be this alternative pathophysiology. What was the initial human disease where investigators started suspecting that some kind of non-biologically living pathogen was involved.
Yeah, I think it was the basis of like non-DNA dependent verification. So it is like, yeah, and it was seen as a big problem for molecular biology because by the time this is the 80s. So in the 80s, like molecular biology has had enormous advances from the 50s to the 80s. Everything was not only from the structure of DNA to the genetic code, uh, to like the ribosomes, to reverse transcriptase. So the structure of molecular biology was almost complete at the time. Like most of the things, most of the basic uh, molecular mechanisms in molecular biology are established. So for something to come and say, okay, this is a DNA, that this is the replication that is not based on nucleic acids, that doesn't involve DNA or RNA or messenger RNA or tRNA or ribosomes or all these things, was very difficult to explain. And that's why initially when it was supposed by recruiters, now it was met with a lot of resistance from the field. But the first human disease that this was used to explain also was the disease called Kuru. It was kind of a shaking neurodegenerative disease that affected the tribal population in Papua New Guinea. Uh, and unfortunately, it was studied by a very bad doctor called Kariem Ezzat. So this guy, who this disease affected mainly children and women in this small tribal population, and he went there and studied the disease in terms of clinical symptoms, and then took brain samples from the uh, deceased kids that had this disease and took it back to the US and injected it into, again, brain homogenate, injecting the brain of these uh, 6 years into chimpanzees, and was able to show that, yeah, after some months, these chimpanzees developed similar symptoms of something like Parkinsonism, like so motor symptoms and also some cognitive symptoms. But unfortunately, and this doesn't get mentioned a lot when mentioning Petrusek. He won the Nobel Prize for this. But he was also in the 90s convicted of child abuse, and he never repented even until his last days. He said that, yeah, this was okay, yeah. Yeah. So this is part of the story that I think, unfortunately, uh, uh, doesn't get told enough, but it is part of this messy scientific history of these diseases. But in the 1990s, there was another disease that was causing a lot of fear. It was the mad cow disease. So again, now something like Scrapie, but now happening cows walking sheep. So cows started to get mad in the sense of like starting to behave weirdly and have also lose weight and have some shaking symptoms. Like this spread across uh, like herds of cows. Um, and there was it was the 90s. The Prion hypothesis have been now postulated for maybe a decade, and since it looked very similar to Scrapie, uh, then this gave a lot of publicity to the Prion hypothesis that it might also explain the spread of spongiform encephalopathy, which what it was called, spongiform encephalopathy, and because it causes like holes in the brain to cow. And so it's led to the slaughter of millions of cows in the UK that were feared to be infected with this spongiform encephalopathy. And it was seen as a prime disease because, again, no virus or bacteria could be found. And then again, if you take the brain homogeneity from one cow, inject it to another, it can induce the disease. So, this homogeneity, it doesn't matter if you boil it or if you use UV irrigations, it can still reduce the disease. So it told people that there is no DNA or RNA involved, and it had to be a prime disease, which means that it's protein only disease, that these proteins propagate and cause other proteins to misfold and lead to the symptoms. And there was fear that the people consuming this meat from this cow would also get the disease. So a lot of surveillance has been established since 1990s to follow up on cases of what was called variant cross-Yacob disease, which is now called acquired pathology, which is also a brain degenerative disease. But fortunately now it's been 30 years since this happened, and in the UK, which was the epicenter of this epidemic, in 30 years, only 200 people have been diagnosed with the variant cross-Yacob disease. Also, millions have consumed this meat during the 90s. So it turned out not to be as dangerous as people thought it is. But again, in the 90s, this caused a lot of, of fear. And in 1997, Dr. Rosener was also awarded the Nobel Prize based on the Prion hypothesis that these diseases are caused by protein-only agents that induce, spread their bad conformation to other proteins in recipient organs.
So the general model that, that's still being taught is that there's these essentially protein conformational templates that are self-propagating, that then we're told something along the lines of it's a protein that then induces this conformational replication and binding to another protein, and then forms the plaques that drive the pathology. Tell us why this has been either unproductive or counterproductive, and then is the evidence on the side that that's what's really happening when we get down to trying to find molecular mechanisms?
Yes, I went into this field based on this knowledge also, because this is what we studied in school. So I didn't start on as like, okay, the final hypothesis, there must be some. I knew that these diseases are untreatable, but I didn't have any bias against it because it made sense at the time. But when I read more, it's like most of these mechanisms that you find in textbooks and in papers, they are cartoons. It's like it's not based on, it's not like the DNA structure, which is based on like real X-ray crystallography of how the molecules are arranged in space in the nucleus and how it works, and it works only in this way. And there's like everything is known about the conformations of the bases and why A can only pair with C and why C can only pair with G, and this is the basis of this preservation of the biological conformation, which is the sequence of the ATC G. Because if this selectivity is not there, if A can pair with G, like then the information will be lost because there have to be some restrictions to make. And this is what Watson and Crick famously ended their 1953 paper was that like it hasn't escaped our notice that this it can be the basis of how templating, how replication of DNA happens. That the arrangement or the sequence of the basis is the actual information, and this restriction of how the splitting can happen only in a certain way is how this information is preserved, not only through replication but also through transcription and translation. So in the 1970s, Crick published a paper, like yeah, I think his most important paper is 1956 on protein synthesis, where he basically foresees the entire framework of molecular biology before it was all found experimentally. So it predicts like mRNA and tRNA and all these molecules that are needed for the nuclear machinery of life to work. And like in 15 years, everything he predicted was found. Uh, so it's amazing. It's a great mind. So looking at this, like so it comes from the structure that was identified by physicists like Louis Whitfield and Rosalind Franklin, and the structure led to the understanding of what is biological information and how the hybridization works and how the targeting works. And so it came from down the top. When you look at the Prion hypothesis, it came the other way. There was no structure to start. It was just a phenomenologic way of understanding, like a kind of something that's difficult to understand. Why using the message to destroy DNA and RNA doesn't destroy the ability of this brain homogeny to transfer infection to recipient RNAs. So that's why in many of the textbooks that we study, we don't see structure. You see, like again, like what you just said, like it's like a tone of this protein, like somehow fishing for another protein to bind into it and change its conformation. But like you studied physics, and I was also trained in physical chemistry and biophysics. This doesn't make any dynamic sense. Why would we do that? What was the driving process of doing that? How this actually happened on the molecular level, and not on the cartoon level, and this what differentiates like real hardcore science from kind of just not very deep understanding of the processes that take place. So my first disillusionment, the Prion hypothesis, was when I was working on a project trying to understand how viruses interact with molecules in biological fluids. So it's something, I come from the nanotechnology field, so there's something in nanotechnology field called the protein corona, where proteins, when you inject a nanoparticle in the bloodstream, for example, there are thousands of proteins in the plasma, and the nanoparticle being like an inert, like it's not a bacteria or something that can prevent these proteins from binding this surface. So what happens is once the nanoparticle enters this biological environment, tens of proteins bind to its surface, and it affects how the nanoparticle distributes in the body and how it delivers whatever cargo it carries and stuff. So there's a lot of literature in the medicine about the protein corona, how to prevent protein corona, how to display protein corona. So I came from this field thinking that yes, viruses are also nanoparticles. They are biologically equivalent to nanoparticles. They are not alive until they are inside cell. So outside the cell, we don't have but interacting with these proteins. So I was studying the protein corona of viruses and how this affects viral infection. That was in 2017. When I came across a paper showing that nanoparticles, so having fixed this this idea that nanoviruses are biophysically equal to nanoparticles, I was reading a paper that how nanoparticles can induce amyloid aggregation. And amyloids are these protein fibrils that are found in diseases like Creutzfeldt-Jakob disease or Alzheimer's disease or Parkinson's disease. And the paper showed that nanoparticles can trigger amyloid aggregation without the presence of any Prion seed or template. So I thought, okay, if nanoparticles can do this, then viruses also can do this because they are the same. And when I had viruses at hand, and I started running these experiments not with nanoparticles but with viruses, I saw that viruses can also induce amyloid aggregation. And this was kind of the whole point of the Prion hypothesis was to say that it is not viruses that are proteins. It has to be a protein on the template. But here I see on the screen of my reader that viruses can very readily do that. So they had the other explanation, and because I cannot dismiss as I see with my own eyes. And when I looked back into the literature, there was no structure except all these cartoons. And so I thought, okay, there is so much unknown here, and so much that the Prion hypothesis can explain. So I thought like that there has to be a reasonable biophysical explanation for what's reported in the literature and what my experiments were telling me that no templating is needed. It has to be a spontaneous process that happens like normal fold, which is also spontaneous process. So normal protein folding based on the intention hypothesis, which says that you don't need the template for protein to fold in its native conformation. It's just the native folding happens spontaneously based on the primary sequence. So what biology needs to do is only to preserve the primary sequence, and then the function automatically is derived from that based on the spontaneous folding that gets its information from the primary sequence. So folding in biology in general is not a templated process. The templating happens only for the nucleic acid sequence information and the translation and translation. But after that, folding is completely non-templated.
See, this is a huge fundamental part of the molecular biology for our audience who is not as familiar with the underlying molecular biology here. This is a really, really, really big sticking point. The reason why DNA is able to have a linear proxy for all of these functional three-dimensional proteins in our bodies. The reason this is a heritable linear sequence that can predict the inherited functional three-dimensional shape of a protein is because of what you just said, that the protein folding is a result of that linear sequence. And
Exactly.
the sort of maybe unacceptable thing that got snuck in as an assumption with the Prion templating concept was that there could be this sort of externally induced conformational templating that was essentially always a cartoon model. Not to pick on the researchers who were invested in this model, but it was essentially a cartoon model that didn't have an underlying molecular explanation. And sort of one of the recurring themes of my podcast is to also remind people you always have to think about the evolutionary biology and the evolutionary implications of a model if we were to pretend that it was true. And the thing that struck me with the old sort of external conformational templating concept with prion disease propagation is that if biology worked this way, it would be profoundly problematic. If you can imagine a world where rather than protein folding being a result of primary base pair sequence, if instead the peptide sequence didn't determine the conformation, if instead proteins were just openly adopting conformational templating from their environment, think of what an absolute train wreck any organism would be that had any form of extracellular fluid environment. Think of what an absolute train wreck our bloodstream would be. Think of it, the train wreck that our cerebrospinal fluid space would be if these spaces would simply have proteins bumping into each other, abandoning their native protein folding and adopting this conformational binding to whatever nearby protein and forming these giant aggregates on a regular basis, if that was the regular way for protein folding to happen, the amount of environmental disruption that would happen for a protein-based living organism, it is almost incomprehensible. And I wish that we could convey an extra step in the reasoning of every single bio paper, every single biomedical paper that gets written. If your model is true, what are the evolutionary implications? And it's something I'll get off my soapbox here, but it's something that I constantly bring up when I'm speaking with researchers in the genetics of lifespan research, aging, and longevity, because when people propose these cartoon models of, we're going to find that one DNA switch that'll make us all live to be 1,000, you always have to ask the question, if this is introducing an evolutionarily high perturbation scenario where a single switch could make this highly perturbable system and a highly perturbable biology, you should probably second guess your model, because if we don't see it acting like it is this easily perturbed as a system, your model might not be right. And now that you're cracking that for prion disease, what did you find out?
No, I really love this point. I mean, uh, we it is very important to think in this way. And again, yeah, if the plasma have thousands of proteins, all this hemoglobin would like change the conformation of albumin, and albumin will change the conformation of whatever, and it will be a mess. And another thing, if a big protein holding would need like uh, if each fold will need its conformation to fold to, then why would the cell buffer replicating DNA in the first place? Have then we would have needed our conformation templates for every single protein that that we have, and we needed these templates would have been needed to split during mitosis and somehow during meiosis and replicate in each cell and preserve these templates like forever, and like it's unfathomable. It's unfathomable. So I think the picture that was nearly complete in the 1970s and 1980s of how I think uh, of how this biological information works, and like the intrinsic hypothesis was the cherry on top of that. But yes, there is like insane amounts of machinery to make sure that this linear sequence of DNA is preserved, is repaired, is uh, faithfully replicated, is faithfully transcribed, is translated. I mean, the amount of machinery that is involved in this process is immense. So, what in short is the uh, scientific of this uh, like like hardcore information that of life, and there is no room simply for other machinery or that has never been seen that ultimately works the other way. And this is the famous triangle that published in 1956 and again in 1970, showing this like the arrow of biological information from, and we published it, this like a version of it recently in our paper also. Like it's like from DNA to RNA and from RNA to protein, but never the other way around. Never from protein to nucleic acid, always from nucleic acid to protein. So because if you want to transfer the information back to nucleic acid, you will require a parallel machinery that completely like reverses a process which doesn't make sense and has never been found. So I think the foldability and spontaneous is very important. And transfer hypothesis, together with the rest, uh, kind of the basic dogma of microbiology, that is DNA to protein, the the information is the sequence information, explain everything in terms of like how information is handled on the molecular level. So there is simply no room for templating in this system. And it's much easier to extend. And this is what we're trying to do now. So if it's not a primary purpose, how this works. You cannot just attack something without like postulating some, like if you think if there is a better explanation, you should come out with it. So this is what we try to do, is that, okay, the protein misfolding is still folding. It is misfolding, but it is folding in a way. So it should still follow the principles of protein folding, which is the sequences. It's just the protein folds depending on the into the most thermodynamically favorable state based on its primary sequence. But this happens only in dilute conditions. But if you use very high concentrations of the protein getting into the supersaturated, then the crowding makes the protein molecules close to each other. So in this molecular proximity at supersaturated condition, the intermolecular hydrogen bond starts to become more favorable than the intramolecular hydrogen bonds, but responsible for mating fold. The intermolecular hydrogen bonds bonding starts to become more favorable. And then instead of the protein folding on itself based on the primary sequence, it starts to make this intermolecular stacking bonds, which leads to the formation of these amyloid fibrils. And since you cannot form intermolecular hydrogen bonds while you are in the helical conformation, because two helical peptides can only come as close as 8 angstroms, which is a big distance to form an intermolecular hydrogen. So the peptide needs to change its conformation to beta sheets, which reduces the molecular distance to 4.8 angstroms, and then the peptides can form this intermolecular hydrogen bonds. I mean, form this long chain of intermolecular hydrogen bonds, one molecule on top of other important another, another molecule forming these fibrils. So this is so the model we try to propose is that at normal concentrations, at normal conditions, the peptide follow the thermodynamic hypothesis of protein folding, and Prieson's law might hold into the 3D structure as important for its function based on its primary sequence. But if you somehow use enormous concentrations or reduce the stability of the peptide, then it starts to interact with itself. The molecule starts to interact with another peptide molecule. And for that, it needs to change its conformation to this generic amyloid conformation, which is sequence independent, by the way. So the information which was preserved and very hardly forced forward with all these elaborate machineries to make sure that the sequence information is preserved, it's suddenly lost to this generic intermolecular interaction, because basically you can push any peptide to form amyloid, and this is something not a lot of people know. So you can form amyloid of insulin, and form amyloid of P53. Like you can form amyloid of like a polyglutamine or polyglutamine or polyart, like any. It is a function of the peptide change. But all you need to do is to have a very high concentration of peptide for it to start forming these intermolecular fibrils. So it's basically a case of peptide crystallization. It has nothing to do with replication in any sense of the word. There is no information transaction going on. The sequence information of the peptide is lost to this generic intermolecular fibrils that they all basically look very similar to each other. The domains that have evolved to specific functions are lost to intermolecular interactions. So instead of kind of a peptide hormone savoring its domains to interact with the receptor, now these domains are involved in these intermolecular interactions in the fibril and are not available to perform its functions, and that's why it is not these fibrils are usually very inert and not very toxic, because most of the domains are sequestered in these intermolecular interactions, and they are not soluble because the domains also, the charges and stuff are not available. So they precipitate our solution. So we think it's just a simple process of protein precipitation. It's very similar to crystallization. And what is the role of virus? Because this is what started me on all of this. And it is, or viruses were circuits, because in that case, viruses do not act as viruses; they act as catalytic surfaces. Because if you study crystallization, you know that there are two different ways of inducing crystal formation. One is called homogeneous nucleation, and another one is called heterogeneous. Now, heterogeneous nucleation is basically if you introduce an exogenous surface into your reaction, the surface acts as a catalyst for forming this first part of the crystal, which is called the seed, which is the kind of the rate limiting step for the phase transition from being a solution at the supersaturated state to first transform into a solid, which is the crystal. So without getting into the details of the classical nucleation theory and how it works and why there is something called the nucleation barrier based on the interfacial energy of creating a new phase within the original solvent phase, but it is very well established in the phase, very the field of crystal over of how this phase transition happens. So there is nothing mysterious about and this is sometimes like we discussed. This is a process that have been known since Gibbs Sapphire. That's more than 100 and maybe 120, 130 years ago. How phase transition happens to molecules, how crystals form. And you could simply apply these principles to proteins at high concentrations. The amyloid phenomenon becomes very explainable on the molecular level without the need to postulate the very cumbersome, as we just discussed, thing of postulating some new templating mechanism.
Can you talk with us about what the difference in our ability to potentially develop therapeutic interventions would be comparing the old model to your model?
This is something that we're also working on. So this view that amyloid, like these proteins, these prions are kind of special type of toxic proteins that go around and transform other proteins to become toxic to the brain. Kind of excluded the idea that these are this is something has to come from the outside, the template from the outside, kind of corrupting the native before that will be creating proteins. Made it the focus on like how these proteins gain the cells, like what we call gain of function. These proteins must be killing the cells by transforming into this vicious type of killer. Like that, you read in the popular science magazines, also like calling them rogue proteins, or like some proteins that turned into toxic proteins, which I think was also cemented by the development of the amyloid cascade hypothesis in the 1990s, which found that these amyloids, like the amyloid beta peptide in Alzheimer's disease, looks also similar like the Prion proteins in Creutzfeldt-Jakob disease. So the idea was cemented that these proteins must be very toxic to the brain, and the therapeutic approach has to be trying to get rid of these toxic proteins. But since this protein, again, any protein can form amyloid, so it would be very evolutionarily very inefficient if all these proteins are made just to be toxic. Again, so these are native proteins that are very evolutionarily conserved. Either the PRP or the amyloid beta or the alpha-synuclein Parkinson, they are all evolutionarily conserved, highly expressed in the brain. So it doesn't make evolutionary sense that these proteins are just there to become toxic and kill active cells. So what we looked at it is systematically. What happens to these proteins is they suddenly disappear from whatever environment they are. If you spark, it's like if you see a crystallization, there are a lot of nice videos of crystallization on YouTube. And what happens is the process, once it's started, just consume the entire soluble populations of molecules available for it. And what happens if you imagine that this is happening in cell, that suddenly the entire population of certain proteins is absent. So, and again, there is a structural reason for this, is because these domains, the domains that were supposed to interact with the receptors or with other proteins, are completely now consumed with these self interactions because of this crystallization has just happened. So we thought that the pathology might be more dependent on the loss of function of this protein rather than again toxic. So this protein losing the initial function is probably more toxic because they are inert, uh, and there, it has been shown a lot that there is this very good correlation, for example, in Alzheimer's disease between the amount of plaques someone has in their brain and the disease progression. So you can find people. This has been shown also post-mortem. You can find people who have very healthy cognition, but you look at the brain after they die, and you find that the brain is filled with amyloid, which is supposed to be, if they are toxic, this person should have had like very, very bad Alzheimer's disease. And this is also seen even with PET scan. Some people, you run PET scan on these people, and you find that they have large amounts of amyloids in the brain, but they are fine. So if these amyloids were toxic, how these people are still fine. So that's why we postulated the loss of function hypothesis, that it doesn't matter because these fibrils are inert and they are not really toxic by themselves. So they probably don't cause toxicity unless they kind of accumulate in a muscle, or like for example, there's a disease called the heart, like the amyloidosis of the heart. So if they accumulate in an organ which has moving parts, the organ becomes stiff, and this is how they cause diseases. But there are no moving parts in the brain, so they can just sit there. But what they do is that they consume the population of soluble functional. So we postulated that yes, it's probably more dependent on the loss of this protein than the gain of function protein from this from the amyloid fibers of the plaques.
Fascinating flip to the entire perspective. And I love the connection to other neurodegenerative diseases because it would be a really big breakthrough insight if we recognize that sometimes the presence of some sort of plaquing phenomenon may not be the driver of the actual pathophysiology, but rather that the depletion of a functionally necessary peptide may actually facilitate the pathophysiology. Because if we assume that the plaque presence is the driver of the disease, 100% of our effort is going to be how do we prevent plaque accumulation, or how would we remove the plaque accumulation. But if it's the depletion of functional proteins, then that would completely flip the perspective on what a therapeutic intervention would even look like.
It would be the complete opposite. You need to increase these proteins, or decrease them. And this is this is what this is why it has been very difficult for us to sometimes publish our papers in the field and stuff, because it sounds crazy. Like you want to increase these proteins, not decrease them, because most of the drug development have been focusing on destroying these, either in the like antibodies or antisense RNA or like CRISPR or anything to remove these proteins. And most of these approaches didn't work, and some of them actually made people worse. And it's very well known in Alzheimer's disease, for example, that in the late 90s, early 2000s, there have been a huge effort in developing the so-called gamma and beta secreted inhibitors, which are supposed to reduce K-beta 42, which is seen as the toxic peptide in Alzheimer's. And in clinical trials, even in healthy volunteers, these drugs worsen, not enhance. So if you use this as an argument also on our side, that, if you reduce this peptides even further, you worsen the condition because the disease is already a loss of function. To kind of test this hypothesis, it's not okay. If we are correct, then this group of patients, no, not patients, this group of people who have plaques in their brains, but they do not have disease, the population is entire. Then if you are correct, then these people must have higher levels of insoluble protein, the soluble peptide compared to the people who have the same amount of plaques, but have the cognitive problems of Alzheimer's. And this is exactly what we found. So we published 2 papers in 2021 and 2022. One is studying around 600 people who have sporadic Alzheimer's disease, and in 2022 in familial Alzheimer's disease. That data were available about the amount of plaques that is by PET scan in these people's brains, and of course their cognitive scores and CSF measurements of the amount of soluble EV42 in their brain. And we found that if you normalize the amount of plaque, what distinguishes someone from having dementia to not having dementia is that the normal cognition have higher levels of the soluble compared to lower levels in the dementia. So it seems that this neuropeptide, EB42, which is the evolutionary concern, it's used all our lives, it's present at higher amounts in healthy individuals in its normal soluble form compared to people with dementia. This is being in its normal physiological concentration is important for cognition. And the disease only proceeds when this peptide goes below a certain level, even if there are no plaques, or even if you have plaques, but you can maintain normal soluble levels of this peptide, you can still maintain good cognition. So based on that, this is what we started this company. It was called Prion Therapeutics to try to replenish this pool of peptides, and now we are also finding other like other pharmacological pathways to increase the level of these peptides and treat it as a loss of function disease, not as a gain of function, because this is the only thing that hasn't been tried in neurodegenerative diseases so far. Because the view has been very focused only on that these peptides are toxic. We need to get rid of them. Like very little about like if they have function or not, what they do. This has been ignored largely, and I think this was a mistake because these proteins are there. They are found in the normal concentration, and so in their normal naked conformation at higher levels in healthy people. So good news, next year we finally are going to start a clinical trial when we're pursuing a molecule that actually increases the levels of EB, not decreases, but increases it in the brain. And it's repurposing. So there has been data about these molecules from previous study that this molecule, which was feared to actually lead to increase in the risk of Alzheimer in the patients taking it, was the retrospective study analyzing the effects of this molecule has shown that the molecule can actually reduce the incidence of AD dramatically in the patients. And we think this is because this molecule has the higher of decreasing AD42. And next year we're testing this molecule in clinical trials and hope to show that maybe the better way to treat these diseases is not like cancer, that there is kind of a toxic mass filling the brain. It's more like diabetes, that there is something that is missing. There is a neuropeptide, a molecule, or like a neurotransmitter that is lost in this aggregation problem, and that while the aggregates, if they are not that toxic, the depletion of this molecule is what's driving the problem.
Thank you so much for taking the time to walk us through both the history of this fascinating disease class, and thank you so much for walking us through this very interesting scientific journey of kind of unpacking the problem with the old way of approaching it and shining some light with your new interesting insights, and hopefully we'll see some therapeutic progress. That is super exciting to see it go from an idea to an intervention. So we'll keep our eyes and ears open for that. And thank you for stopping by the podcast.
Thank you very much, Matt.
Mm-hmm.